Hello everyone, welcome to the virtual class for Biotechnology, Principles and Processes. Now first of all, we must understand the meaning of biotechnology. It is basically relates to the techniques of using living organisms or enzymes or any other products from the organisms to produce products and processes which are useful to human, and that includes like in vitro fertilization to have test tube baby, to address the problems of infertility, then synthesizing genes and using it for various beneficial effects, for improving the plant characteristics, and also developing DNA vaccine and correcting a defective gene, and thereby to address the problems of genetic disorders in human being. Now, according to European Federation of Biotechnology, the definition of biotechnology is it is the integration of natural science and organisms, cells and parts thereof, molecular analogues, and for products and services. So this encompass a large field, and in this chapter we will discuss basically the principles of biotechnology. Now the basic principle of biotechnology are: one is the genetic engineering. Genetic engineering means to modify the DNA of any target organism, thereby changing the phenotype of the organism along with its genotype. Another one is bioprocess engineering. That means it maintains the sterile conditions to support the growth of the large quantities of desired microbes and other eukaryotic cells, which are used for production of various new or modified products like enzymes, antibiotics, vaccines, etc. Now, the techniques of genetic engineering involves basically it is nothing but combining the desired genes which is obtained or isolated from a select organisms, and to create a recombinant DNA. That means the desired genes are combined with a vector DNA to form the recombinant DNA. Then it follows the transmission of the genes and then gene cloning and host DNA maintenance. So the fundamental steps of the genetic engineering include to find the DNA which contains the desired genes, then injecting the host with the identified DNA, and preserving the introduced DNA in the host and transmission of the DNA to the offspring. But to obtain this three steps, there are several steps in the background. Now, before we deal with those steps, quickly we just go through some definitions which are very commonly used in the field of biotechnology. So, number one is, what is the fate of DNA when it is transferred to the cell of an alien organism? Is it possible for the DNA to multiply itself in the progeny cells of the organism? No. Normally, it cannot directly multiply when it is inserted into the progeny cells or the target cells. When it gets integrated into the genome of the recipient, it may multiply and may be inherited along with the host DNA. So this is because the alien piece of DNA has become a part of the chromosome in the host cell, in the recipient cell, and which has the ability to replicate. And in a chromosome, there is a specific DNA sequence which is responsible for initiating the replication process. And this particular sequence of DNA in a chromosome is called origin of replication, or ORI. Therefore, for multiplication of any alien piece of DNA in an organism, it needs to be a part of the chromosome having a specific sequence of ORI. Now, an alien DNA is linked with the origin of replication, fine, so that the alien piece of DNA will now replicate and multiply itself in the host organism. As a result, it produces a multiple identical copies of any template DNA. template DNA is the desired DNA. And this process of getting the multiple identical copies of the template DNA is called cloning. Another term which is very commonly used is plasmid. We know that plasmids are autonomously replicating circular extra chromosomal DNA of a prokaryotic cell in a bacterium especially. And plasmid is a very useful vector for recombinant DNA technology, which we will learn in our next few slides. Since we cannot directly transfer the desired DNA into the recipient DNA, we need an intermediate into which we incorporate the desired DNA, and then this desired DNA is incorporated easily into the plasmid, and that forms a recombinant DNA, and that recombinant plasmid can be easily transmitted into a host cell for multiplication and further processing. Now, construction of first artificial recombinant DNA, it was a milestone in the field of biotechnology. This was accomplished by Stanley Cohen and Herbert Boyer in 1972. They isolated antibiotic resistant gene by cutting out a piece of DNA from a plasmid. Now, this was achieved by linking a gene which codes for antibiotic resistance property with a native plasmid of Salmonella typhi. So what they did was the required gene which they wanted to incorporate through the recombinant DNA technology was cut with a restriction enzyme from a host DNA molecule. And then plasmid was obtained from the isolated bacterial cell, which was also cut with the help of the same restriction enzyme. And then this desired DNA, the template DNA, was added with this plasmid with the help of the enzyme called ligase, and we get a recombinant DNA, half plasmid and half template DNA. This is the recombinant DNA which was then inserted into a bacterial cell where it was allowed to multiply, because as the bacterial cell multiplies by binary fission, this recombinant DNA also gets multiplied. So it go on multiplying, and therefore it can produce several copies of that identical recombinant DNA. When this, as a result, this antibiotic resistant gene in E. coli was called cloning of antibiotic resistant gene. So what are the tools of recombinant DNA technology? First one is we learned about restriction enzymes, then polymerase enzymes, ligases. These three are different groups of enzymes having different functions, vectors, and competent host organism. Now, very common vectors which are used in biotechnology. One is, as I already told, about plasmid because of the circular shape, and it is an autonomously replicating or extracellular DNA. So it is it can be cut by the restriction endonuclease in any in a particular point, and then the DNA template can be added by the enzyme ligase. Similar vector can also be bacteriophage, because bacteriophage is a virus having DNA as their genetic material, and this DNA can also be used for making recombinant DNA, and phage can be very easily introduced into a host cell, into a bacterial cell, where this recombinant DNA will enter into the bacterial cell and it will grow there. It will be incorporated with the bacterial genome, and then it will continue to produce more recombinant DNA products. So first tool we shall discuss is the restriction enzymes. Now, restriction enzymes are called biological scissors. Two enzymes from E. coli were obtained which were responsible for restricting the growth of bacteriophage, and they were isolated long back in 1963. One of them added a methyl group to DNA, and the other cut DNA into segments. Now, the enzymes which cut DNA intermediately, that means in between, not at the extreme poles, it is the endonuclease. So restriction enzymes are either exonuclease or endonuclease. Exonuclease are those which cuts the DNA from the, if this is the DNA, this cuts from a pole, and endonuclease will cut the DNA into two segments from the middle or internally. So the first restriction endonuclease isolated was named HIND2. It was found that they always cut DNA molecules at a particular point by recognizing a specific sequence of 6 base pairs known as recognition sequence. After that, a number of restriction enzymes have been isolated, having their which can recognize different recognition sequences. Now, the very commonly used restriction enzyme endonuclease is one is EcoRI, and we must learn the nomenclature of the restriction enzymes. EcoRI, this name, the first letter is derived from the genus name. That means EcoRI comes from the genus Escherichia coli RY13. So, and the next two letters are coming from the species name. So, E stands from Escherichia and CO coming from coli, CO, of that prokaryotic cell. And the Roman number after the name show the order in which the enzymes were isolated from the bacterial strain. It was R was the bacterial, RY was the strain, so it was R. And then accordingly it was 1, 2, accordingly there are 245 restriction endonuclease enzymes isolated from Escherichia coli. Now this naming of restriction endonuclease enzyme is important, and this can be illustrated with the example of EcoRI. Now, as we have already discussed, this exonuclease, they remove nucleotides from the ends, the terminal ends, but endonuclease cut at a specific position within the DNA. And each restriction endonuclease recognizes a specific palindromic nucleotide sequence in DNA. Now, palindrome, as we know by dictionary, is a sequence of words which read same on the both the sides. So in case of DNA, palindrome in DNA is a sequence of base pairs that reads same on two strands when orientation of reading is kept same. For example, in a DNA strand from 5' to 3', if the base sequence is GAATTC, then its complementary strand will have, for G it is C, for A it is T, so it will read as CTTAAG. And now when we read from the opposite sides, so we find that from in both the strands, from the 5' end, it reads the same. So this is a palindromic sequence in case of DNA. This illustrates the action of the restriction enzyme EcoRI. Now EcoRI cuts the DNA between the bases G and A when the sequence GAATTC. So this is the sequence, palindromic sequence responsible for the enzyme EcoR1. Now this red one is the vector DNA and this black one is the foreign DNA, means the template DNA from where the fragments will be taken. Now the restriction endonuclease cuts in both the cases, EcoR1 cuts at the same palindromic DNA sequence, and they form a sticky end. That means the segments are not cut smoothly. Sticky ends are formed so that one helix is a little longer than its contemporary. This is because the sticky end of the foreign DNA will be bonded with the sticky end of the vector DNA by complementary base pairing, and that will result in the formation of the recombinant DNA. So this cutting, the sticky end portion is important, which is necessary for the sticky ends actually is responsible for the complementary, help to bind the bases, complementary bases, and that gives the recombinant DNA. Now, if we go to the mechanism of action, this illustrates more clearly. We can find that the cleavage side is GA. So here it is, the cleavage side is GA, and on this side it is GA. So that is why it will be cleaved here and this will be cleaved there. So there will be a longer end here and that will be a longer end there, and that will be complementary bonded with to form the double helix of the recombinant DNA. So restriction enzyme cuts the strand of the DNA a little away from the center. This single-stranded portion leaves the single-stranded portions at the end, and the overhanging stretches are called the sticky ends because they form hydrogen bonds with their complementary cut counterpart. This hydrogen bonding part, that means the stickiness of the ends, facilitates the action of the enzyme DNA ligase for joining. So restriction endonucleases are used in genetic engineering to form the recombinant molecules, and the sticky ends are complementary to each other when cut by the same restriction enzyme, and therefore they can be joined by the DNA ligases. So the two basic enzymes which are used for this making this recombinant DNA, first is restriction endonuclease, another one is DNA ligase, and the third important point is the presence of the sticky ends, which facilitates the function of ligase and also help to form the hydrogen bonds with the complementary counterparts. Now, once the DNA, recombinant DNA is obtained, then the cutting of the DNA, before obtaining the recombinant DNA, when the first DNA is cut by the restriction endonucleases, results in the fragments of DNA. Now, the technique which separates DNA fragments based on their sizes, it is called gel electrophoresis, the process. Now, DNA fragments are negatively charged molecules and they can be separated by forcing them to move towards the anode under an electric field through a medium or matrix which is formed by the agarose gel. Now, agarose is a natural polymer. It's a gel-like substance which is obtained from the seaweeds, and this agarose is spread in a electrophoretic tray, and then it is allowed to solidify there, and then the DNA samples are applied at the point, at the slots are there, at each slot the DNAs are applied, and then when it is connected to the electric field, the molecules start moving. The largest one, they precipitate earlier, and the smaller one, they can travel further distances. As a result, the DNA bands are separated like this. Now, the separated DNA fragments can be visualized under UV radiation, and for this reason, they are stained by a substance called ethidium bromide. So, in the gel electrophoresis, what we must remember is these are the features which should be remembered. DNA fragments being negatively charged, they will move from anode under the electric field in a matrix. The matrix is essentially made up of agarose, a natural polymer obtained from seaweed. Once the DNA molecules are separated according to their sizes, smaller molecules will move further, and the separated DNA fragments will be visualized by staining with a compound called ethidium bromide, and which will be visualized only under UV radiation. The DNA fragments will be seen as a bright orange colored bands, and these separated bands can be cut from the gel matrix, and this is called elution. This eluted DNA, it is a purified also, because it is this particular DNA will be occupying a band based on its molecular weight only, and so it is not containing any other associated molecules in it. So this purified DNA fragments can be used in constructing the recombinant DNA by joining them with the cloning vector. So before joining with the ligase, the DNA which is cut from the desired organism, it must be separated, isolated and purified with the help of gel electrophoresis technique. Next, we come to the cloning vectors. Now, cloning vectors are the DNA molecules which can carry a foreign DNA segment into the host cell, and such vectors are very useful in recombinant DNA technology, as we have already discussed. One is the plasmid, being it's a circular extra chromosomal DNA which can autonomously replicate in a bacterial cell, or a bacteriophage virus which carry the DNA molecule, and when it infects the bacteria, only the DNA enters into the host bacterial cell, and that DNA, the viral DNA containing the recombinant DNA, can easily incorporate with the bacterial genome and continue to multiply, producing multiple copies. So there are cosmid hybrid vectors which are derived from the plasmids and bacteria. If an alien piece of DNA is linked with bacteriophage or plasmid DNA, its number can be multiplied equal to the copy number of the plasmid or bacteriophage. Now here we can see the E. coli cloning vector, which is termed as PBR322. This is a very important diagram. This shows the plasmid structure, and there are the common features which are required to facilitate the cloning of into the vector are: one is the ORI, as we have discussed, or origin of replication. Then this restriction enzyme which will cut at the point, that is EcoR1, this part. Then selectable markers. Selectable markers are those resistant genes. Here we find ampicillin resistant and tetracycline resistant genes, and vectors for cloning genes in the plants and animals. So this is the origin of replication, from where the replication will start. So any piece of DNA when linked to this sequence can be made to replicate in the host cell, and once it is multiplied, replicated in the host cell, it can be easily identified by the selectable marker. This sequence, this ORI, is also responsible for controlling the copy number of the linked DNA. So origin of replication has two functions of ORI. It's important. Now we are coming to selectable markers. Selectable markers are helpful for identifying and eliminating the recombinants, that is the transformants, from the non-transformants, that is the non-recombinants. Transformation is a process through which a piece of DNA is introduced into a host bacterium. The genes which codes for their resistance to antibiotics such as ampicillin, tetracycline, chloramphenicol are useful markers for E. coli. The ligation of alien DNA is carried out at a restriction site which is present in one of the two antibiotic resistance gene. That means this, if we join the template DNA in this portion, then originally present this resistant gene, for example, if it is ampicillin resistant gene, this will be cut from the vector plasmid, and the new DNA will be incorporated in this region to obtain the recombinant DNA. So the recombinant DNA will not carry the ampicillin resistant portion, that is selectable marker 1 maybe. So the recombinant plasmids will lose either the ampicillin resistant gene or tetracycline resistant gene due to the insertion of the foreign DNA. But it still can be selected out from the non-recombinant ones by plating the transformants on ampicillin containing medium. If the ligation is made at, for example, this is a tetracycline resistant gene, then ampicillin gene is present there. So if we join new template DNA in this portion, then the recombinant DNA will not have this tetracycline resistant gene, but it will be containing the ampicillin resistant gene. So when this recombinant organism, the plasmids, they are the transformants, the cells with the recombinant plasmids, they are allowed to grow on ampicillin medium. The transformants growing on the ampicillin containing the medium are then transferred on a medium containing tetracycline. So recombinants will grow in ampicillin containing medium because they still have the ampicillin resistant gene, but they will not continue to grow in the tetracycline containing medium, because now they do not have the tetracycline resistance gene. So their growth in the tetracycline medium is not possible. By this selectable markers are therefore used to identify whether the transformation has taken place correctly or not. And the non-recombinants, if both the resistant genes are present, they will grow on the medium containing both the antibiotic. So selection of recombinants due to inactivation of antibiotic is a cumbersome process. So alternative selectable markers are also developed, and this is normally done with the basis to produce color in presence of a chromogenic substance, means a colored substrate. In this method, a recombinant DNA is inserted with coding sequence of an enzyme called J3-galactosidase. This results into inactivation of the enzyme P-galactosidase. So the bacterial colonies whose plasmids do not have an insert, they will continue to produce the blue color, but others which do not produce any color when grown on a chromogenic substrate, that will help to identify the recombinant from the non-recombinants. Now again we are coming back to this plasmid structure to find out the cloning sites. Cloning sites are required to link the alien DNA with the vector. So the vectors requires very few or single recognition sites. There may be one or more than one recognition sites within the vector. Accordingly, the template DNA can be added onto them. The vectors which are used in this genetic engineering, there are different types of vectors. One is cloning vector, which is very common, as we have already found an example, PBR322, then bacteriophage. So this is a cloning vector. And another one is expression vector. Now in plants, the tumor-inducing plasmid of Agrobacterium tumeficans, which is a bacteria, is used as a cloning vector. Agrobacterium tumeficans is actually a pathogen for several dicot plants. It affects the dicot plants and cause some diseases. During infection, it delivers a piece of DNA known as tDNA in the TI plasmid, which transforms the normal plant cells into tumor cells to produce the chemicals which are required by the pathogens, and thereby it causes the disease. Now, retrovirus, adenovirus, papilloma viruses are also known to be used as cloning vectors, and this because of their ability to transform normal cells into cancerous cells. Now, this tDNA, TY plasmid, has the capacity to to convert the normal plant cells into tumor cells. This property is actually utilized when this tDNA, tumor-inducing plasmid of this agrobacterium, is used as a vector. This capacity helps in producing multiple copies, that is cloning process. Next, we are discussing the competent host organisms. Now, who are competent? Competent means the organisms which can be readily transformed with the help of recombinant DNA. It is competent host organism is required because DNA, being a hydrophilic molecule, cannot pass through cell membranes. So the bacteria should be made competent to accept the DNA molecule. And this competency is the ability of the cell to take up foreign DNA. And methods to make a cell competent are maybe two types. One is by chemical transformation, by incubating the bacterial cell into certain chemicals and maintaining a definite temperature, giving a heat shock. The cell membrane then allows the DNA molecules to enter, and after recovery, we can get this host organism with the recombinant DNA. Another technique is electroporation. Same thing is the cell is first incubated, and then instead of heat shock, electric shock is given. As a result, the recombinant DNA molecules, they are allowed to enter into the host organism to produce the and the host cell then recover and continues to multiply, producing multiple copies. In the chemical method, the chemical which is used is, it is a divalent cation like calcium, which increases the pore size in the cell wall, and then it is given heat shock. Means first the DNA is kept on ice, and then suddenly the temperature is increased to 42 degrees centigrade, and this sudden shock breaks the cell wall and enables the bacteria to take up the recombinant DNA. And in physical methods, recombinant DNA is directly injected into the nucleus of an animal cell by microinjection method. In plant cells, the cells are bombarded with high velocity microparticles of inert elements like gold or tungsten, which are coated with DNA, and they are called the gene gun method, ballistic method. And the disarmed pathogen vectors, when allowed to infect the cell, they can transfer the recombinant DNA into the host. Now we will discuss a special technique in the biotechnology, which is polymerase chain reaction, PCR. It is the process for producing multiple copies of the gene of interest, which is synthesized in vitro. And for this, the requirements are two sets of primers. The two sets of primers are nothing but small chemically synthesized oligonucleotides, which are complementary to the regions of the template DNA. This is the one requisition, and another is the DNA polymerase, and third item which is required are the nucleotides, that is the deoxyribonucleotides, which are provided in the reaction tube. So the three aspects necessary to perform PCR: number one, two sets of primers, which are nothing but oligonucleotides. Oligonucleotides are short chain nucleotides, which should be complementary to the selected regions of DNA. So the primers will depend upon the DNA template which we want to incorporate in the genetic engineering process. The enzyme DNA polymerase is necessary to continue the process of multiplication, and the enzyme extends the primers using the nucleotides. Nucleotides, as we know, they are four types for synthesis of DNA, containing adenine, guanine, cytosine, and thymine, which are provided in the reaction medium, and the genomic DNA as the template DNA. Genomic DNA means which will multiply on the basis of these primers. So if this process is replicated several times, the single segment of DNA can be amplified billion times within a very small duration. Now, this particular one point which we should remember is this particular enzyme, DNA polymerase, is a special enzyme which is thermostable. And this thermostable DNA polymerase was isolated from a bacterium called Thermus aquaticus, and hence this DNA polymerase is named as Taq polymerase. This remain active during high temperature induced denaturation process. Now, the technique of PCR, it involves three steps. Now, in this figure, we can see this red marked region is the region to be amplified. So, this is the first double-stranded DNA. This double-stranded DNA, the two strands are separated by the process of denaturation, where it is heated. By applying heat, these two strands are separated. And on these separated strands, the primers are attached. This process is called annealing. So where the primers get attached to the respective DNA strands based on the complementary basis. And with the help of these primers, the region which we want to be amplified, they start amplifying with the help of the Taq polymerase and deoxynucleotides, which are present in the reaction tube. So we can see that this amplification is continuing, and the two daughter DNA strands, double helix, are gradually extending, and the third phase is therefore called extension. And if we can set the cycles in 30 cycles, then it will produce about 1 billion copies. And this is a computerized technique, obviously, and this whole thing, this preparation of the sample, everything before setting the tubes into the cycle, everything must be performed under sterile condition. So amplified fragment, if desired, can now be used to ligate with a vector for further cloning. if we want to clone it further. So this is a basic technique for PCR. Now, at the end, we can summarize the process steps for recombinant technology, DNA technology. The first point is isolation of the genetic material. That is the template DNA, the genome. So the genetic material depends upon from the which cells we want to isolate. So we have to treat the donor cells with the enzymes accordingly. If it is from plant cell, then the enzyme used will be cellulase. If it is from fungal cell, it will be chitinase, because the fungal cell wall is made up of chitin. And if it is bacteria, then the enzyme used will be lysozyme. And other than these three, DNA is often associated with other macromolecules like RNA, protease, lipase, etc. So accordingly, other enzymes can be used also to remove proteins. We use protease, lipase for lipids, and RNase for removing RNA. After isolating the genetic material from the desired cell, selected cell, then it is necessary to cut the DNA, template DNA, the genomic DNA, at specific locations by restriction endonuclease. And at the same time, the plasmid DNA, that is the vector DNA, is also cut. If it is plasmid, then vector DNA is also cut at specific locations by same restriction endonuclease. After that, the amplification of gene of interest will be done using polymerase chain reaction. So cutting of genomic DNA at specific locations by restriction endonuclease, then amplification of gene of interest using PCR. After that, insertion of the recombinant DNA into the host organism. For obtaining this recombinant DNA, we introduce this genomic DNA into the vector. In the host organism, we obtain the foreign gene product, that is the recombinant protein. Now, if we want to produce the recombinant product in large scale, then this laboratory scale methods are not very fruitful. So the whole process is performed in bioreactors. Bioreactors are vessels in which raw materials are biologically converted into specific products, individual enzymes, using microbial plant, animal, or human cells. And a bioreactor provides the optimal conditions for achieving the desired product by providing optimum growth conditions, temperature, substrate, salts, vitamins, oxygen, just mimicking the environment of a living cell. And then afterwards, these products are processed further by separation and purification to obtain the finished product. And then that can be used for various applications in biotechnology.